go for a meeting at 11 o'clock. Rajesh, Shukla and Vikas, no, please start because you have to go to a meeting with Vice Chancellor at 11 o'clock. So you have to finish the webinar at 11 o'clock. Please start the meeting.
Sure. Sure.
Very good morning to everybody. And I, Professor Vikas Pandey, Dean Faculty of Pharmaceutical Science on behalf of Mangalayatan University, Jabalpur, welcomes you all esteemed guests and participants on today's groundbreaking webinar on novel composite materials in the trace reaction of arsenic and some pharmaceuticals, a laboratory to device development. Today we embark on an extraordinary journey by acquiring fruitful knowledge on nano.
विकास जी आपका वॉइस ब्रेक हो रहा है, थोड़ा नजदीक में रखो माइक.
It's a matter of pride and honor for me and Mangalayatan Institute Jabalpur to welcome over today this team.
आपका वॉइस ब्रेक हो रहा है विकास जी.
Diwakar Tiwari sir, who is Professor, Department of Chemistry, Mizoram Central University, Director of DRDO-Industry, Academy Center of Excellence, Mizoram University. His research wise in excellence is itself an inspiration for all of us. Firstly, I would like to share a very interesting thing that it's a matter of pride for Jabalpur that sir has completed his post-graduation MSc from Rani Durgavati Vishwavidyalaya, Jabalpur in 1988. Further education, doctorate from BHU, Varanasi in 1990. A vast experience of research in the area of environmental chemistry and advanced analytical chemistry. Several national and international, including two SIA New Delhi, STF fellow twice, visiting researcher Chinese Academy of visiting Professor Wang Dong University, expert member leader education committee for RE Center or in the Council of Dr. Academic Council of University of Punjab, University, Board of Study of platform will be open time for question and advice.
Thank you. Shall I start? Actually, voice is not.
Yes.
Yeah.
Yes, sir. His voice is not clear, but your voice is okay.
Yes, it's fine, sir. No problem. Thank you, sir, for giving me this opportunity. We are definitely. I am very delighted delivering a lecture in Jabalpur in Sanskar Dhani, where I born and brought up. I will say in Jabalpur.
He's a Jabalpurian. Diwakar sir is a Jabalpurian.
Yes, sir. Thank you, sir. Thank you very much, sir.
I graduated from Science College, and now I came to know that it is a modern science college and like a autonomous college. So it's really I'm delighted delivering a lecture in Jabalpur. Thank you, sir, for giving me this opportunity to deliver lecture. In fact, I was thinking to ask you to give me opportunity to deliver a lecture in your university at Mangalayatan.
I know you are a seasoned scientist of Mizoram University, and so for everybody's information, Diwakar Tiwari is one of the eminent scientists of Mizoram University, and he has published more than 175 papers with high impact Scopus papers. And every year he visits Korea as a visiting scientist for 2 months. And he's having a lot of startups and innovations he developed in Mizoram University. We are very lucky to have a collaboration with our university, and also his expertise to our Mangalayatan University will be very much useful, and we will have collaboration with him in future also, because he comes very regularly to Jabalpur. So we are very much happy, sir, for your kind acceptance and gracing this webinar.
Thank you, sir. Thank you. I'm thankful, in fact, sir, as well as all the faculty and the Mangalayatan University at large. So thank you, sir, for giving me this opportunity. And good morning to everyone. And I would like to just share my slides. If you allow me.
Yes, sir, you can share, sir.
Sir, the permission required, it seems, sir.
Sir, I just promote the police permit, Diwakar Tiwari. Okay, sir, he has permitted you.
Okay. It's showing, sir, you must grant permission in order to share, sir. It's showing.
No, he just, he's.
Pramod ji,
kar diya?
Update.
Permit
Okay,
kiya?
otherwise, sir, I will go with this. Just 1 minute, sir.
He has given, sir. Otherwise, you can send to us. I will arrange to share slides.
Just 1 minute, sir. I'll try with some other.
You try, sir. He has permitted you, sir. कर दिया सर, देखो सर एक बार.
Yeah.
Good morning, sir. Ravikant here.
Yes, good morning, sir.
Namaskaram, sir.
Namaste, sir.
Good morning, sir.
Good morning, everyone.
Are you able to get it, sir, Diwakar, sir?
Diwakar, sir, your mic is off. Your mic is off, sir.
It is coming.
Yes.
It has come, sir, now. Just, Diwakar, sir, it has come.
I think it's visible, sir.
Yes, sir, it is visible. It is coming.
Yes, sir.
Okay,
Yes, sir.
thank you, sir. Thank you. So, as already, thank you for giving me this opportunity and for kind introduction, sir. So, I'll be talking about that novel materials in the trace detection of arsenic and some pharmaceuticals, a laboratory to the device development. As sir already mentioned, that I'm also looking after this as the director for the DISV, DRDO-Industry Academia Center of Excellence. This is the only center in the northeastern region. So this is a brief view. The department of the chemistry on the left hand side. Right hand side, this is the panoramic view. I will say that the of the Mizoram University is on the hilltop, and the left hand side is School of the Physical Sciences and some of our this is the below to that the building, our auditorium and all that. I'll not go in that detail. This is one of the paper which I published in 2012. But 2017, in the Applied Clay Science, this was the top cited article in this journal. Just briefly, I would like to mention. And the other paper, which was published 2000, long back 2012, and this was the top cited article in Chemical Engineering Journal. And this is the impact factor of this journal is now is 15. So these are some of the papers, as that I am taking care as the editor-in-chief of this UGC care listed journal, that is the Science and Technology Journal. The other journal, which is Environmental Engineering Research, the associate editor of this journal, having impact of almost 4. I'll start with this very pertinent. I used to mention like this what Albert Einstein, he just wrote to his son once, that life is like riding a bicycle. To keep your balance, you must keep moving. So this is very pertinent in fact, in our whatever the endeavors that we are having. The other I used to share with my students, especially that which the Swami Vivekananda he mentioned once, that in a day when you don't come across any problem, you can be sure that you are traveling in a wrong path. So what happens usually, students, they come with some problems, or they say that what the problem has been given, they are not getting what the results what they are expecting. So I used to tell that now you are moving in the right direction if you are not getting, because this is the research. In research, always you will be facing the problem. So these are the things that I used to share with my students. I would like to share with this August gathering. So the contents of my presentation is the motivation, how we have been motivated in this type of the research. Then the introduction, materials, methods, what we have done, results and discussion, and followed by a brief conclusions. So arsenic, if I talk about the arsenic, the report which was published way back in Science, October 1996, that is the India's spreading health crisis draws the global arsenic experts. That was mentioned in that the deadly trail, 7 districts in West Bengal have been hit hard by the arsenic poisoning from tube wells along with the India-Bangladesh, the border. So this arsenic problem, if you see on the right-hand side, that no, all this, the red mark, this is the water, the groundwater, it is contaminated, extensively contaminated with the arsenic, and the people are the society what they are lying there, they are too much affected with this. This is one of the picture which I have taken, and if you try to see here, the entire, this is the mapping that we could get it, and this is through the the website only we could get it, and you can see that the arsenic contamination, or the water which is contaminated with the arsenic, it is large part of the country, it is the arsenic is being detected, and all that level of course maybe it may vary, but the arsenic has been detected in the large part. In the northeastern part also, the part of the Assam and all, it is too much affected with the arsenic. And when we are doing some kind of the research also here, also we have observed that the part of this, the Arunachal Pradesh and the Assam and all, lot of arsenic along with the iron and the fluoride contamination is the predominant there. So of course one of the project also I am working on that, the device development related and all that, or in the remediation also. The arsenic pathway, it is a very common. You can see that both the anthropogenic or the natural sources are these are the 2 ways by which arsenic is entering into the environment, whether it is the aquatic environment or in the air and all that. This is a simple pathway which you can just see that the fertilizers and the pesticides, the volcanoes and all that, through that the arsenic will be coming, and then through this the water is the recharge of this water, and again it is recycled and all that, the spring water, through that it will be coming. So all this the arsenic pathway that you can see that. The other part of what I would be discussing today, that is related to the micropollutants. When we talk about the micropollutants, but before that I would like to mention one of the report which was published in the India Today, 29th September 2015, where they have they have shown the concern about the endocrine disrupting chemicals, that we call it the EDCs, and particularly they were talking about the bisphenol A, how this bisphenol A entering into the water and how it is going to affect and how it is going to be as the EDC, one of the potential, or I will say that one of the concern about the human health and all. So when we talk about in general, when we talk about the micropollutant, I will tell you what are the micropollutants and what are the concern about the micropollutants. But when we see this, the BPA or the endocrine disrupting chemicals, the concerns are infertility and kind of the hormones related cancers and neurological issues and all that. And this is all related not only with the BPA, but the other kind of the pollutants are also there, and that is called as the micropollutant. Micropollutants, I will say that it is a generalized term that is being used. That includes not only the the EDCs, but the pharmaceuticals and other kind of the pollutants are also there. The question was there, why do we call it the micropollutants and all that? A simple definition also it has been given by the EP and all that. The micropollutants are organic or the mineral contaminants or anthropogenic or the natural origin that can be harmful to the human health as well as the aquatic environment. They can be found at very low concentrations in water and are generated from industrial processes, pharmaceuticals manufactured for human use and veterinary drugs, personal hygienic products, industrial or household chemicals, detergents, cosmetics, textiles, pesticides, or from microsubstances in coatings or the paints. This is a general definition. The general, the broad, I will say that the types of the micropollutants are there. But the question was that why do we call it the micropollutants and how they are entering into the aquatic environment or in general in the environment. So this was the, if you try to see that this is a very simple picture which I can show here, that the simple treatment plant, what we are having in this simple treatment plant, there are the 2-3 units are there. The first is the primary sedimentation tank is there, followed by the aeration tank along with the microbial activity, and the secondary treatment, that biological treatment that we try to do it, followed by the chlorination, by this microbial activity has been removed. So these are the simple way by which we try to treat our water. But if you have you ever seen that what is the effluent of this, the treated water? What is the quality of this water, the treated water? What is the quality of the influent, and what is the quality of the effluent? So the treated water, when you try to monitor it, then you will find that kind of the chemicals which are coming out through this treated water at the very low level, somewhere around microgram to the nanogram level, and that is being, creating the problem. And the developed countries, for example, the US or the Korea and the other countries, they are having a monitoring system of the treated water also. They used to monitor it, and large number of, I will say that a large number of the pollutants they try to, or large number of the compounds they try to identify and try to detect it, and it has been detected that a large number of the pollutants are being detected there, and that needs I will say that the advanced treatment is required to remove all that. For example, I give an example. Suppose the antibiotics are coming, say the pharmaceutical type of the compounds are coming out. As we all know that when we are taking, for say, the antibiotics, when we are taking it, antibiotics is not completely metabolized in the biological system. Large quantity of these antibiotics are coming out through the feces or through the urine and all that. And once they enter through the sewage treatment plant and all that, and there, if you try to see that it is not completely being removed or degraded, and then what happens, they are coming into the effluent water, and which you are, it will be mixing with the aquatic environment or the water bodies, and water bodies also slowly is contaminated with the micropollutants. And then what happens, if the, say for example, the antibiotics are coming into, then that is once the water is being, people are taking that, the contaminated water, slowly this what you say, that the immunity that is going to be there to decrease, and then when the antibiotics are being given or injected or given to the person, that will not be that effective. So that is the simple reason that these micropollutants need to be identified and need to be treated effectively. So if you look at that, the US EPA, the definition, they say that an exogenous agent that interferes with the synthesis, secretion, transport, binding action, or elimination of the natural hormones in the body that are responsible for the maintenance of homeostasis, reproduction, development, and or behavior. So that is called as the micropollutants, as I mentioned, that microgram to the nanogram level that need to be detected. So the detection system also, we need an efficient detection system by which you can detect these micropollutants at very low level. So this is the micropollutants, the pathway and all that, whether it is the residential area or it is the healthcare system, through this the municipal or the sewage waste water is coming and the sewage treatment facility is there, or through this the septic tanks are there and then aquifer are contaminated, or through this and all this, once it is treated, the sludge or the water, the treated water, when they mix with the water bodies, say treated effluent or agricultural runoff, then again it is contaminating the soil or the water bodies. So these are the pathways by which the micropollutants are being, the moved or they are distributed in the aquatic or the soil. These are the micropollutants in groundwater, a grave concern. But if you look at that, this is in the country also, some of the reports that has been published where they have shown their concern about the micropollutants in the water bodies. And when I was looking into the literature also, I have seen that some of the studies or some of the, So these kind of the screening, they have mapping, they have done it. And in Hyderabad also, when I was looking, there are the 5-6 different the pharmaceuticals are being detected there. And if you look at that, the Punjab side, there are also some of the reports they have reported. But at this stage, what I have observed that not much reports have been published, or not the much data has been collected, so that by which we can have a proper the mapping of the country, that the water or the reservoirs or our rivers or our the groundwater, what is the contamination level of these micropollutants and all. So one of the article which was published, and it says this, how to make sure your water is safe to drink when you are pregnant. So this is in which they have very much emphasized that when you are hydrating yourself, you are hydrating the two persons, in fact. So this is the status of the groundwater pollution due to pharmaceuticals. When we too much concerned about the pharmaceuticals or the different kind of the pharmaceuticals, this is the global, the mapping, where you can see that the India, it is having, which is not the entire thing are being mapped, but if you try to see that, even then 31 to the 100, the pharmaceuticals are being detected in the country. And this white marks are showing that these are the data is not available. So as I mentioned, that the Hyderabad to be the polluted with 12 different types of the pharmaceuticals, the major ones being citrazine, ciprofloxacin, enoxacin, and so on. So these are some of the literature which we can get it about the groundwater which is polluted with the pharmaceuticals compounds. So with this, the background, we were thinking that why not to develop the techniques by which, as we all know that for detecting all these micropollutants, we need a very sophisticated instrumentation. For example, for the arsenic and all that, ICP-MS and all that, we have to use it. And as we know that in the drinking water or in the aquatic environment, the permissible level is somewhere it is the 50 ppb. So we have to detect, our detection system must be much lower than that. So we wanted that a device has to be developed which is which can detect much below to that level. So we tried with developing different kind of the materials, and that materials we try to use it for the detection system in the electrochemical detection system. For that, we had a good collaboration with the Korean university, and the device also being developed based on that. So basically what we have done, we have utilized the natural clay. As you all know that the clay these are the phyllosilicates, are all these are the tetrahedral or the octahedral tetrahedral silicate or the octahedral aluminate are being arranged, and through that, in between that, there are the exchangeable cations, and these exchangeable cations we are trying to introduce the long chain organic compounds, and then we use to modify the materials. For example, the HDTMA and all that, we try to introduce within that the galleries and all, or within that the interlayer, and then we try to prop up the layers, and then we try to utilize it for the detection system. So these are the simple methodology that we have utilized it. We have fabricated the electrode. I will tell you that how we have fabricated the electrode, and then we have introduced in for the detection system. Initially, we have done in the laboratory level, as I mentioned, that from the laboratory to the detection system that we have done it, or the device development that we have done it. So here what we have done, we have done the fabrication at the laboratory, the electrodes, and that electrodes will be giving you the better sensitivity and all that. So that and sensitivity and selectivity, these are the two important aspects that need to be addressed. So that we have done it with the help of the electrodes in the electrochemical system. So this is one of the paper which was published in the EER, and what we have done, the schematic of the sensor studies, as I mentioned that the fabrication of the electrode was one of the aspect that we wanted to do it. So for that, the glassy carbon electrode that we have used it. Here is the Teflon tube. The Teflon tube was 4 centimeter long. In that, the paste which we have made it with the help of this our material, along with the carbon powder, paraffin oil, to make it a good paste. And then this paste has been introduced inside this, or I can say that packed with this Teflon tube, and the titanium wire has been introduced there to make the electrical connectivity. And then we have introduced in the right hand side, you can see that these are the the electrochemical system or the cyclic voltammetry and all. This is a full the electrochemical system. With that, we try to analyze the analytes and all that. These are the material characterization that we have done. That is the SEM images along with the EDX data on the that heterogeneous structure, and then the once this the organic or the SGTMA has been introduced, that is the surfactant, when it has been introduced, you can see that on the surface this has been aggregated, and this is going to exchange the exchangeable cations, and because of that the D value has been slightly increased. That on the XRD data that you can get it from 1.499, it has increased to the 1.563. Slight increase was there, and with the IR data we have confirmed that the organic molecule has been introduced. Another aspect in terms of the electrochemical characterization of the materials or the electrode is that the important thing is that the RCT value, that is the RCT is nothing but the charge transfer resistance. The charge transfer, lower the charge transfer resistance, better the the electrochemical or the exchange, the process that can take place. So this is the Nyquist plot on the left hand side, and this is semicircle of the Nyquist, and with that we have optimized the RCT value. RCT value when the carbon paste electrode was there, it was 6,342 ohms, but that has been decreased to 3,000, almost half of the value that has been decreased. And the semicircle also, you can see that it is significantly decreased. So that what we can say that the charge transfer reactions at the electrode surface that can easily can take place, and the better sensitivity one can get it. The other aspect of that cyclic voltammogram of the arsenic 3 that we obtained at the different pH, and we found that the lower pH is better in detection system, because the current what we have observed for the oxidation or the reduction of this arsenic 3, that was much enhanced at the lower pH. So with these the basic data, what we try to do, the calibration line, we try to obtain it varying the concentration of the arsenic 3 and what we could get it, and through that the LOD value that we try to obtain it, and the LOD value is significantly low. LOD value that we could get it, that is 0.0036 microgram per liter, that is almost the ppb level. So as we know that the 50 ppb is the permissible level, but we could get with the help of this the electrochemical system in the laboratory, of course it is the 0.0036 the ppb level. And these are some interference and the real water the analysis that we could get. We have done it with the real water. What we have done, we have collected some of the water from the river, and then we analyze the river water, and after that we have spiked with the known concentration of the arsenic, and we could get a very good the straight line and all that. And even then, even the LOD value was also very low as comparable to that. So this is the another paper which was published in the Journal of the Electroanalytical Chemistry. Here what we have done, the similar material we have used for the detection of the cadmium and the lead in the the two simultaneous detection of the cadmium and the lead, that we have done it. Now I will come on the other part, that is the detection of arsenic, of course, by using the slightly different material. What we have done here, where we have we have introduced of course the bentonite, but the bentonite is being grafted with the silane, and a specific silane, that is the trichlorooctadecyl silane, which is the structure has been given here. We have grafted it, and we could get the nanocomposite material. And then what we have done, we have decorated this nanocomposite with the silver and the gold nanoparticles. Silver and the gold nanoparticles that we have obtained using this the natural the phytochemicals which has been obtained by the guava leaves, and it's a very simple process that we have adopted. That means that the leaves that we have taken at very not very high temperature, we have agitated it and slightly raised the temperature somewhere around 70 to 80 degrees C, and then the extract has been collected, and that extract has been used to reduce the silver and the gold the precursors, the ions, and that has been converted to silver and the gold nanoparticles, or in the colloidal form that has been formed, and the color has been changed. That the detection and all, the characterization and all that I will be showing here. At the same time, the phytochemicals test for the qualitative test that we have conducted for the different compounds that like the alkaloids, tannins, saponins, and all that. That was the positive and all. So this is the process, and then we have decorated our nanocomposite material which was previously prepared. That is the the silane grafted bentonite and then decorated with the silver and the gold. So we will be getting the two different materials. One is based on the silver, the other one is based on the gold. And then extensively, we have characterized these materials, but at the same time, now this time what we have done, instead of the glassy carbon electrode, what we have done, instead of the carbon paste electrode, this time we have used the glassy carbon electrode. The glassy carbon electrode, the surface of this glassy carbon has been modified with the help of these our materials. So what we did, we have using these nanocomposites with this, we have mixed with the DMF and deionized water. In that, we have dissolved it, polished the prior to this, the coating of this, the material, we have polished the surface, and then by using the drop casting method, we have casted the surface of this glassy carbon electrode. This is a simple process which you can see that, and then we have utilized it for the detection purpose now. So these are the characterization of our materials. Very quickly, I will be going that the silver and the gold characteristic peaks by the UV visible spectra that we could get it on the. If you try to see that the picture on the in the inset where we have shown the yellow color of the silver colloidal particles on the right hand side, the gold nanoparticles are being shown here in the liquid form, and we have taken the UV visible, and these are the characteristic spectra. And then what we have done, the FTIR spectra we have obtained for these materials. Through this, the data, only thing that we can confirm that the organic silane has been introduced, so that has been marked here. The organic silane is being introduced. Then we could get the same images of these materials, the bare glassy carbon. What we have done, we have taken a similar the glassy carbon sheet, and that glassy carbon sheet has been analyzed for the, we have exposed it for the same analysis, the image, and then you can see that a very smooth surface of the glassy carbon is being obtained. And on the surface of this glassy carbon, we have introduced the bentonite, or the TCBN, that is the composite material, and the silver and the gold based material. You can see that the silver and the gold nanoparticles are well seen on the surface of this, and it is a very heterogeneous surface that we can see on this. So then EDX data, with that EDX data, we have confirmed that the silver and the gold peaks that's in the spectra that we could get it, that confirmed that the silver and gold is being impregnated or that has been decorated on the surface. So these are the 10 images we could get it, and through that we could obtain that, and we have confirmed with the XRD data also that the silver nanoparticles or the gold nanoparticles is being aggregated, and the size, nanoparticle size, somewhere around 20 to 25 nanometers is being average particle size that was there. So at the same time, we have done the the AFM. So with the AFM data, we could get the surface roughness, and the surface roughness is an important aspect. So 17.6 nanometers for the silver-based material, and then 11.9 nanometers for the gold-based material that we could get it with the help of this the atomic force microscopic data. So the electrochemical characterization that we have done, so the two ways by which we have done the electrochemical characterization. The one way is that the scan rate studies, we are exciting the electrode by the increasing the potential at a different the time intervals, and then the scan that is called as the scan rate. So its scan rate was increased from the 50 to 200 millivolts per second, and using this Randall-Savick equation, we could get the electroactive surface area. On the right hand side, you can see this the EIS measurement that we have conducted, the impedance spectroscopy. So through that, we try to get this the RCT value, which is something related to the mass transfer reaction that is taking place at the surface, using this the Randall circuit or this the equivalent circuits. Through this data, what we have observed that electroactive surface area was for the bare glassy carbon electrode, which was 0.269 into 10 to the power of -3. Silver and gold materials are somewhere around 5 to 6, means that almost 3 times the surface area, electroactive surface area has been increased. With the help of this Nyquist plot, we could get the RCT value, which was for the bare glassy carbon electrode, which was 16.53 ohms, which has been decreased to 3.13 and 3.22. That is a significant decrease, almost the 5 times decrease of the the charge transfer reaction, charge transfer resistance. At the same time, the RS, the solution resistance, also we can see that most of these almost the similar values we could get it. So this is the detection of the arsenic 3 and arsenic 5. Those who are working on the arsenic 3 and arsenic 5, as we all know that arsenic usually in the two oxidation states. The one is the arsenic 3 oxidation state, arsenic 5 oxidation. Most of the electrochemical studies, when you look at that, they have done arsenic 3, but arsenic 5, very few reports you will obtain directly the detection. I have not seen direct detection of the arsenic 5. One paper earlier also we published for the detection of arsenic 5. Here what we did, we tried using these materials, the detection of arsenic 3 and arsenic 5, and you can see that on the left hand side the arsenic 3 is detected, on the right hand side arsenic 5 is being detected. Cyclic voltammogram is also being shown at the bottom, and here you can see that the arsenic 3, a very low pH, very low potential, it's being giving a good peak. On the right hand side the arsenic 5, little bit higher potential, it will be giving a very sharp peak. So with that, the LOD of this detection of arsenic 3 and arsenic 5 was obtained, and it is the 0.037 ppb and 0.087 ppb of the arsenic 5 that we could achieve with the help of these material, these fabricated electrodes. The question was that can we use it for the real water? So real water that we have collected the Chitte real water, and with that we try to, here again we could get a very good straight line, the calibration line. We could obtain it for varying the concentration from 10 ppb to the 40 ppb, 50 ppb. And this is on the right hand side the water quality that has been measured, and that has been shown here. Inorganic carbon, of course, of this water was very high. And the second thing is that these kind of the studies, those who are doing, they must be knowing that the percentage of the recovery is an important quantity. So recovery, we could obtain it from 94 to the 105%, which is a very reasonable. And most of the studies, if we will be getting from more than 90% to 110%, this is a good recovery that we used to say. So similarly for the arsenic 5, we have conducted for the real water, and here again a good recovery that we have obtained, and a good calibration line with the R square of 0.9947 that we could get it. So there was a real challenge for detecting these two, arsenic 3 and arsenic 5. And I will say that this is one of the things that we have conducted, that arsenic 3 and arsenic 5, I have not seen directly detection of these two elements, these two oxidation states of the arsenic, and that we have done it with the help, in the simultaneous detection in the real water also that we have done, and both are detected very nicely. And we can see that good calibration line is being obtained, and the two different peaks for the arsenic 3 and arsenic 5 has been observed, and recovery percentage was also very high. And then I'll go very quickly on the pharmaceuticals, the two different pharmaceuticals, like the, especially the antibiotics, like the sulfamethoxazole and sulfamethoxine, which has been published in the two different journals, that is ECS, the Electrochemical Society. The other one is the Microchemical Journal. Already their data has been published. These two materials has been used for the detection of these two. And the cyclic voltammogram shows that this is kind of the irreversible process. Sulfamethoxazole or sulfamethoxine is being oxidized but not reduced. That's why a single peak has been observed on the oxidation site. And we have taken the advantage of that, and when we did the pH dependence data, where we could see that the pH, the 4 to 5 pH is an optimum pH for detecting these two pharmaceutical compounds. And then using the silver and the gold based material, we try to detect this sulfamethoxazole, and we have seen that with the help of this, the LOD, a good calibration line is being obtained, and LOD was 0.022 milligram per liter, that is the ppm level, and for the gold based material we got 0.036 ppm level. Similarly for the sulfamethoxine we have done, and good calibration line here also we could get it, and here the silver and the gold based material we could get the LOD, that is the limit of detection, is 0.027 and 0.035 ppm level. These are the coexisting ions. Some of the ions, particularly like the copper and all, they have hampered the detection, otherwise not significantly hampered. These are the studies that we have done, the surface runoff water that we have used it for the sulfamethoxazole, and then the recovery was also very high recovery we could get it. That means that the real water is being spiked with the known concentration of the sulfamethoxazole. And then again we have conducted the water quality also, it has been mentioned on the right hand side. Here again the nitrates, the sulfates and other quantities, especially this inorganic carbon is relatively high, or the non-purgeable organic carbon is also relatively high. And based on that, the data has been created. So this is the spring water for the sulfamethoxine. Here again, the recovery was very high even in the spring water sample. So these are some of the data that we could get it. And this is the one of the device which has been developed. So when we have completed this arsenic 3 detection, and then what we have done, they have asked us to collaborate with the industry and develop the device. So we have collaborated one of the industry, and then this is a small device. You can see it's not very big. It is already commercialized, and this device has been used on the front. You can see this is the small micro electrodes. This is the carbon based electrode that we have fabricated, and this electrodes directly can be used, and in this the monitor directly the arsenic concentration can be detected. So this is the small device, a very portable device which you can take it into the field or wherever the water you want to measure, and you can detect what is the arsenic, the contamination level is there, and the detection limit is also very low, much below to the 50 ppb. Okay, so based on this, the studies, the device has been developed. So these are the pictures of the Aizol. I will say the Aizol city, the aerial view of the Aizol city. On the right hand side, we had one workshop here in Mizoram University, and the DGLS, DGTM, Director General of Technology Management or Life Sciences, along with the directors, all they have visited, and we had a very good gathering in Mizoram University. And at the same time, I would like to thank the two my research scholars. The one is the Lalmit Swami and the Melody Lalshai, who have contributed significantly doing this work. So, sir, thank you very much. Thank you for giving me this opportunity.
Thank you very much, sir. And I think thank you very much, sir. And we have some questions related to this webinar, sir. I got in my personal number. So the question is, actually they wanted to know what is the traditional technique for the detecting of arsenic trivalent and pentavalent form of impurities in any pharmaceutical compounds other than the electrochemical.
There are the traditional way by which usually what they do, if you want at the very low level, naturally you have to go for this AS or the ICP-MS. These are the two ways. Otherwise, on-site detection, they used to do some kind of the colorimetric methods and all that. They are trying to use it based on some kind of complexation and all that. So I have not, for this, the other methods we have not gone through.
Yes, sir. So everybody has known, sir, the pollutant had a strong bonding, means they are polar in nature. So, and stable also with any other molecules. So how to rectify the impurities from any other components by using the electrochemical method?
See, the basis of the electrochemical method, we have to understand first. The basis is that any compound, unless until it is oxidized or reduced at the surface of the electrode at an applied optimum potential, is difficult to detect. So why this arsenic 5 could not be detected? The reason was that the arsenic 5 requires very high potential to, again, whether you want further oxidation is difficult. But of course, you can reduce it to the arsenic 3 or arsenic 0, and then again you oxidize it. In a cyclic voltammetry, that will be possible. But when we will be introducing a different kind of the surfaces by which at an optimum potential this can be detected. So this is the one aspect that we have to keep in mind, that whenever we are trying to, for example, the pharmaceuticals, when I was talking about, say the sulfamethoxazole or sulfamethoxine. So they have been oxidized, and once they are oxidized, they are not again recombined, isn't it? So it will be this kind of the irreversible process. So irreversible means once the oxidized, some different compound is being formed and again cannot be, cannot form. So in the cyclic voltammetry, of course, the oxidation peak is being seen, but in the reductive peak, there is no such reduction is taking place. So this is one thing that we have to keep into mind. And if it is complex also, for example, arsenic is, say, for example, iron is there. We are talking about if it is in the complex form. In the inorganic chemistry also, we try to use to see that what is the oxidation state of the iron in the complex species. With the help of this, the cyclic voltammetry. So this is a simple way that the oxidation state, whether it is complex species also, that can.
Sir, good morning. One question from my side, sir. This is Dr. Ravikant.
Sorry, I think Professor Pandey also talking something.
Oh, okay, okay.
No. Sir,
Yes.
please, Ravikant sir, please carry on. Please.
Yeah, good morning, sir. This is Professor Ravikant from Mangalayatan University, Aligarh campus. I am looking after the research department here. Sir, one question from my side. Since I am working as a in area of organoarsenic compound, sir.
Okay, sir.
So, can we detect organoarsenic 3 and organoarsenic 5 compound by this method, sir?
Definitely.
By this,
Definitely.
Okay. Thank you so much, sir. Thank you so much, sir.
Thank you, sir, Professor Ravikant sir, for joining this webinar, today webinar with us. And really, sir, Professor Diwakar sir, today's webinar is a very intellectual with, I think, for research candidate and students we are involved in research in analytical chemistry. So you are using the simultaneous estimation of traces, characterize the traces element like arsenic, iron, or lead, whatever the traces element by using the nanocomposite material. So I think it's a very grateful time for researchers. After all, this is Dr. Rajesh Shukla, Professor and Head Department of Pharmacy. I would like to heartily thanks to today's guest and speaker, respected Dr. Diwakar sir, who is currently working at currently working at Director DRDO-Industry Academia Center of Excellence, Mizoram University. University. It is a great privilege that the respected sir joined us today webinar, who gave a fantastic and informative session and addressed on scientific topic, novel nanocomposite material and trace detection of arsenic. Please, interesting topic, novel nanocomposite material in the trace detection of arsenic from pharmaceuticals as a laboratory to device development. So, nanocomposite area is known the nanocomposite material that have a solid structure. Structure in which the distance between the phases is list-wise form of dimension and nanoscale size of and general form of an inorganic matrix set. So, on the set of this organic phase that may be used in trace detection for many impurities in pharmaceutical studies, such as behavioral and learning and memory. We thank you very much, sir. Thank you to all students of Pharmacy, Forensic Agriculture Department of Mangalayatan University and students who participated outside the university for the attentive during the webinar. And also, I would like to thank to respected VC sir, Mangalayatan University, Jabalpur, Professor K.R. Sambasiwara sir, Professor Pro VC ma'am, Vrita Karusaluja ma'am, and our registrar of Mangalayatan University, Jabalpur, Dr. V.S. Kishore sir, who is inspiring us and encouraging to conduct this webinar and made us take it further. So last but not least, at the end of webinar, I would like to thank everyone who joined the webinar, my colleagues, academicians, and dear students. Again, and also request today speaker, respected Dr. Diwakar Tiwari sir, to continue giving such wonderful and informative session webinar in the future. Dhanyavad sir, bahut bahut dhanyavad.
And sir, we sincerely welcome you sir to Jabalpur.
Yeah,
To visit
sure.
your native state.
Sure. Okay,
जबलपुर
sir.
अवश्य पधारें सर.
जरूर.
VC sir, Saluja ma'am, VC sir, Saluja ma'am.
Thank you, sir. Thank you, Diwakar ji.
Thank you, sir.
Thank you very much. Thank you, sir.
Okay, sir. I'm leaving, sir.
And yes, sir. You have a meeting at 11 o'clock.
Yes,
You
sir.
mentioned.
Yes, sir. Okay, sir. Okay,
Okay,
sir.
sir.
Very,
I'll
very
talk
thank
to
you,
you
sir.
later.
Vikas, sir. Thank you,
Yes,
sir.
Thanks.
क्या था कि माइक ऑन रहता है हमारा तुम्हारा तो
इको करता है।
इको करता है आवाज हमारी की आवाज लाइन से तो वो बोल रहा था कि विकास अब शायद बात कर रहे हैं कुछ मुझसे मतलब ऐसा दिल्ली में सब फिर आपको
कौन बोल रहा है?
दिवाकर बोला